All right, guys, we are back. Okay. Let me get this all pumped in here.
Did you send out the connect code?
I did. They're in your email. Check your ATA email.
The connect code. I didn't get it.
You should have gotten it. It's in your
I
ATA
did.
email.
Huh?
In your ATA email.
The only thing I got was the slides.
Check again. I just sent them out.
Okay, it came through at 12:50.
Yeah. Come on, man. All right. All right.
And Mr. Richard,
Yes, ma'am.
I have my access code,
Yeah.
but for some reason I can't use it. It's something I can't get in.
Okay. What browser are you using, and are you using a computer?
Chrome.
Use Chrome on a laptop, and it should take you in. Make sure you type in, make sure that you know whether you're typing in letters and numbers.
I signed in already, but I
Yeah.
don't know where to put the access code.
Well, so what you'll probably have to do, well, so you're in, can you see the assignments?
No.
Well, then back all the way out and close out your browser, and then go back into Connect, and then it should ask you for a login. Okay. So what we're going to do today is we're going to talk about different ways to identify bacteria so that we know what they are, so that we know how to treat the infections that they cause. Okay, so let's get in to share here, share. That. And we got a new share. That. All right. So the idea here is that we have to use tools under our control that allow us to identify what it is that's making you sick. Okay. So what you're doing is playing detective, microbe detective, using these different techniques. Okay. Sometimes these techniques will rely on the genetic information that comes from the microbe. Okay, and so we might actually look at the DNA for a clue. All right, a lot of modern techniques use that. Sometimes we'll rely on staining the bacteria and looking at them under the microscope to help us identify, like what shape are they? How are they arranged? All right. And then we may look at their ability to live on different kinds of media to determine what they are. And so some bacteria will grow on some media and some won't. And that helps us narrow down the possibilities. Okay. And then once we get to that point, we might use an antibiotic sensitivity test to determine which antibiotic is going to be most effective at controlling the microbe. Okay. So those are essentially a series of steps that you do in a clinical lab after you take what you call a culture, right? You might take a throat culture or a skin culture, and then you'll grow it up, and then you'll analyze what grows and try to determine what's in there, and then figure out from what's in there what it is that's making the patient sick. Okay. And that allows you to pick an antibiotic that's going to be effective against knocking down what's causing the disease while allowing your other organ systems to take it easy so they don't have to work so hard clearing out the drug, right? You don't want to give a drug that you have to give in such a high dose that actually the dosage of the drug takes a toll on things like your liver and your kidneys and so forth, which are responsible for clearing breakdown products from those drugs out of your system. Okay. So we identify bacteria in a sample from a patient based on their shape, right? The shape of the colonies, the color of the colonies, the texture of the colonies, and those would be growing on media, or their microscopic shape and arrangement, right? What do the cells look like? Do they look like little BBs? Do they look like rods? Do they look like spirochetes? Okay. And are they even bacteria, right? You know, sometimes you're going to get an infection with something like a protozoan or even a worm or a virus, right? And that falls outside of a lot of the techniques that we would use to identify bacteria, right? And then we can look at their immunologic characteristics, meaning what? What molecules do they have on their surface? And the way we figure that out is by throwing antibodies that only attach to certain molecules at them. And once they cling to the surface and generate either a colored reaction from a dye that's attached to them or an enzyme that's attached to them, we know that that molecule is there and we have an idea of what that pathogen is. And then genotypic, which is where we actually get the genetic material from the bacteria and we examine that directly to tell us what it is. Okay. So those are different methods. And sometimes we combine them, right? We can use the genetic identification technique as a catch-all, a save-all, okay? The problem with a lot of genetic techniques is that they can be easily contaminated, okay? So they can be challenging to do correctly. The other techniques that we're going to talk about are still very valuable, okay? We can also use tomography and MRI and PET as ways to look at certain types of infections, okay? But the stuff we're going to do actually in lab is going to go back to these old-fashioned kind of methods where we determine what grows on what kind of media, the texture and the arrangement and the appearance of the colonies, okay, and whether or not they grow even, and then looking at the cells themselves and seeing what they're shaped like and what they stain with, okay? And then we can even do biochemical tests to determine what sort of enzymes they have, all right? So a light microscope allows us to see what the cells are shaped like, how big they are, and how they're arranged. Are they in chains? Are they in clusters? Are they in pairs? Okay. We can do a gram stain and an acid fast stain to determine the makeup of the cell wall, right? And that tells us a lot about the bacteria right away. Then we can check for endospores, granules, and capsules using those stain techniques as well. A lot of those are negative stains where you stain the background, but you don't stain the structure, but you can see an outline of the structure. Electron microscopy is much more intensive and expensive, okay? And it allows us to see things like the cell wall and flagella and pili and fimbriae. And that's not something we generally use in a clinical setting. That's more for what we would call an applied or a basic science laboratory, where we're trying to really get in depth about a particular microbe and understand at the molecular level how it works, okay? So that's something that really won't be addressed too much in what you guys do. Traits that you can see with the naked eye are the colony characteristics, right? When they grow on media, what color are they? What media do they grow on? What is their texture? Are they fuzzy? Are they shiny? Are they dull looking? Okay. How are they arranged? How big are they? Okay. And that is reflected in how quickly they grow as well. And we look at their pattern of growth in broth and gelatin media. Okay. And the reason for that is that lets us determine what their oxygen requirements are if we're looking at broth. Okay. And in gelatin media, we can look at something called motility, meaning do they have the ability to move through the media using things like flagella, okay? Back before we had genetic methods, we would use traditional tests like we're using in lab here to figure out what kind of bacteria were making you sick, okay? So we ask questions like, what sort of enzymes do they have? And what other biochemical properties do they show? Okay. A lot of tests exist to pick out a particular enzyme or to determine what it is the bacteria eat in order to thrive. Right. That tells us a lot about what they are. Right. So we can kind of with here, we can make some notes, right? Okay. So we could say, you know, what chemical reactions can they do? Okay. And then down here, what do they need to grow? Okay. So to do that, we look at things like fermentation of sugars, ability to digest certain molecules, generation of certain gases, and whether or not they're sensitive to certain antibiotics, like in the Kirby-Bauer test that we sort of worked with last week. Okay. If we look at specific substances, we can also help to identify the microbe. We can look at what the cell wall is made of, and we can look at membrane and lipid profiles using something called a mass spec, all right, which is again beyond the scope of what you're going to be doing in the lab, on a clinical floor. Okay, a mass spec actually cracks pieces of the structure down into tiny fragments and then is able to determine their shape and their size, okay, and match that to data that they already have to figure out what sort of a molecule that is exactly, okay. But this, the peptide and composition of the cell wall, you would get through staining. Genotypic methods, to make a long story short, just isolate genetic material and compare sequence information. Okay. So we look at the sequence of DNA that we are able to isolate from the patient, and we compare that to information that we already have from other organisms, and we see where there's a match. Okay. And one of the techniques that helps us do this is something called PCR, which allows us to amplify DNA. Meaning what? Make a whole lot of copies of DNA from a tiny, tiny amount of the DNA that you get from your sample. And then we can take that DNA and we can find out its sequence, all right, and compare that to information we already have. The same kind of stuff they do at crime scenes. So think of it like crime scene DNA testing, where you don't need much to find out whether or not that particular organism is there or not, okay? Immunologic methods use antibodies, okay? All right, so the antibody will be attached to a dye or enzyme that makes a color. You can see the and analyze. Sorry. Right. It's a lot easier than testing for the microbe itself because, again, it's very sensitive. Which means you don't need much for the test to do its work. And we've got kits available for a lot of different pathogens. Examples would be things like C. diff, E. coli, okay, and on and on, okay? All right. We're going to look at factors that affect the ID of infectious agent from a sample and look at the different kinds of tests we can use, okay? So important in identifying the bad guy in your patient is how the specimens are collected, handled, stored, and cultured, right? aseptic technique is imperative, right? Aseptic technique is designed to reduce contamination. Okay. Reduce contamination. And the idea there, right, if you reduce contamination, is that you'll be looking at what's in your sample and not what's in your lab, okay? Sterile sample containers and tools prevent that. You need to take care that samples contain only the microbes from the patient. So only the infected site is sampled and not anything else, okay? In a sputum sample, contamination with saliva should be avoided because there's a lot of bacteria in it, okay? Throat and nasopharyngeal samples, the swab shouldn't touch the tongue, cheeks, or the saliva, okay? Occasionally, saliva samples are needed for dental diagnosis, and we get those by having the patient spit into a container. In a urine sample, we take it aseptically from the bladder with a catheter, or we can do a clean catch where we wash the external urethra and collect the urine midstream, right? So the idea is that the beginning of urination will be contaminated but midstream is clean. And by clean, you mean doesn't contain bacteria that come from healthy tissue. There's bacteria that might cause a UTI, say. Any contaminating microbes from the clean catch method can be differentiated from pathogens relatively easily. The dirty catch is the first urine that is needed for some samples. The mucus lining of the urethra, vagina, or cervix can be sampled with a swab or an applicator. For skin samples, skin can be swabbed or scraped with a scalpel to get to the deeper layers. And wounds are cleaned before swabbing for culture to avoid collecting normal bacteria in the area, okay? Sterile material includes blood, CSF, and tissue fluid that are removed by sterile needle. Antisepsis of the puncture site is important, right? So usually an alcohol wipe, all right, to avoid a lipid embolus, and then maybe some betadine, okay? Other sources of specimens are the ear canal, nasal cavity, okay, and tissue that's been removed surgically. We have to have a proper collection technique, transport technique, and storage technique, right? You don't want the sample to begin to decay, right? Quicker than normal. And you don't want the sample contaminated. You have to label and ID the specimens and provide an accurate patient history to get good results, meaningful results. Patient analysis for signs of infection include things like fever and mucus production. In collection and analysis of the specimen, direct testing using microscopic, immunologic, or genetic methods is beneficial. Cultivation, isolation, and ID of pathogens generally follows, okay? Presumptive data are placing the isolated microbe into a preliminary category, and confirmatory data nail down what it is that's making you sick. The results that are entered into a summary patient chart that can be used in assessment and treatment is important so that the doctor is able to select the right drug. to knock the infection down. Okay. To understand and communicate lab results is important in successful treatment, right? Some diseases are diagnosed without looking at the actual living microbe, right? We can use serum tests to provide indirect evidence for specific pathogens through the antibodies that they create, okay? So the antibodies can report on current antibodies can report on current or past infection. Remember that because of memory, we can generate memory cells that stay with us our whole life and continue to produce antibody even after the pathogen and its antigen are gone from the body. So this is evidence that you've been hit before. Skin testing is important in identifying people who have had exposure to things like rubella and TB, right? So these are called sensitivity tests. Where we hit you with an antigen under epidermal layer. Look for induration. I'd say, what's induration? It's a hard knot that can form after you apply the skin test, okay? And it forms because antibody is moving in where the injection took place in an attempt to defend you, okay? Some pathogens are ID'd solely on signs and symptoms because it's difficult for us to isolate and stain and grow them, okay? These are called fastidious pathogens. What you're looking at here is just a typical lab sheet and the kind of things that you would fill out, okay? Once you're able to get material from the sample, grow it, and then run a series of tests on it to try and identify what's in there, okay? So you can see here, for instance, what's growing in the culture, right? What drugs it's sensitive to or resistant to, okay? Whether there's acid-fast bacteria in there, whether there's fungus in there, okay? And whether there's hidden blood in the stool called occult blood, right? So those are just a few of the pieces of information that you can get, okay? We talk about PCR and talk about how we use that information in order to determine what sort of pathogen might be inside you. And we'll talk about ribosomal RNA, okay? So when we do that, all right, let's see here, biochemical testing. Zip, zip, zip. There's hybridization. Did we get to that? Okay. All right. So when we talk about these old phenotypic methods, we're looking at the shape and arrangement of cells when we use the microscope, okay? So we can use the gram or the acid-fast stain to tell us something about the cell wall. So what is the cell wall in this pathogen? There's just a couple of examples, right? In specialized media, we can enrich for a particular pathogen, even if it's there in small numbers, or if it gets easily overgrown by other bacteria. In selective media, we have non-sterile specimens that have a lot of different bacteria in them, and this encourages the growth of only the pathogen, okay, and suppresses everything else. Okay, so this is a way to filter out contamination. By selection. Okay. In differential media, what happens is that different types of bacteria look different on the same media. All right, they might have different colors, they might have different appearance, okay, but they'll all grow. A pure culture has to be gotten before isolation media is used so that the other steps are going to be accurate, right? In biochemical testing, we ask the question, what molecules are on the surface or inside, in some cases, the bacteria, all right? And so we look for things like enzymes by checking to see whether the products of those chemical reactions are there, all right? So they can be tricked into producing a color change using certain special dyes, right? You culture the microbe in a media with this special dye in it, and then you see if the dye indicates that that product is there, okay? Microbial expression of the enzyme is visible by the presence or the absence of a colored dye or the appearance of the surrounding media, okay? No colorization usually means that the enzyme for using that particular dye isn't there, which means that the bacteria doesn't fall into that group, okay? Some tests include carbohydrate fermentation, where we generate an acid or a gas, right? So we usually have a color indicator that checks for pH, right? Or a color indicator that checks for things like oxygen or CO2, okay? Or sometimes even ammonia, right? The ability to hydrolyze gelatin, starch, and other polymers puts you in a certain group, as well as the presence of catalase, oxidase, and coagulase, okay? Which means that you are either, if you have these, aerobic or aerotolerant, meaning you either need the oxygen to survive or you can put up with the oxygen. Other products can be detected by rapid, tiny systems that pick up to 23 characteristics. And you can see some examples of those systems here, right? Unknown microbe and different substrates. This is going to be their negative control. And then these other ones indicate whether or not there's a bad guy in there, okay, if there is a color change. In a dichotomous key, we look at one characteristic and determine, based on the presence or absence, which way on the key we should go, okay? So for microbes, that includes things like their shape, their arrangement, their gram staining characteristics, okay? Just to name a few. And the flow charts are going to trace a route of ID by offering pairs of different characteristics. Okay. Eventually, we get to an endpoint, and the name of the genus and species for that combination of characteristics is going to show up. Okay. The tables allow you more complete information and are preferred by a lot of laboratories today, even over genetic testing. Other types of tests include phage typing. Now you might say, okay, what's that all about? Well, remember that we have in the environment these little pathogens of bacteria called bacteriophage that only latch on to typical specific bacterial species. And when they do, they create a plaque, right? Because the cells in that area either rupture and release the phage, or they grow so slowly that they generate a round area of slower growth, right? So we take a lawn of bacterial cells, we grow them up on solid media, we put a grid of blocks on the bottom of the plate, and then clear areas are going to be lysed cells, which indicate sensitivity to phage. Then based on that particular phage's host range, we know who the bad guy is, okay? Susceptible animals are needed to cultivate Mycobacterium and Treponema, okay? Well, bird embryos are used to grow host cell dependent Rickettsia, chlamydia, and virus, okay? And that's usually a chick embryo. Antimicrobial sensitivity tests are important in determining which drugs we're going to use to treat. Also useful is presumptive identification of strep, Pseudomonas, and Clostridium, because we have a lot of established ways of dealing with those infections. Okay. It's critical to find out if an isolate from a specimen is clinically important or if it is a contaminant, okay? Some colonies of E. coli in a urine sample can be normal, but several hundred means a UTI, okay? A single colony of a true pathogen, such as Mycobacterium tuberculosis, or an opportunist in a sterile site, is suggestive of disease, okay? Related isolation and repeated isolation of a pure culture of microorganisms can mean that it is the candidate to cause the sickness, okay? We'll talk about PCR, talk about hybridization, and talk about rRNA analysis, okay? All right, so what is PCR? All right, PCR is just a way to take a tiny amount of DNA from a sample and then using enzymes and DNA building blocks and a device called a thermocycler, make large amounts of particular stretches of DNA from a very small amount that's in the source. And this is called amplification, okay? Amplification. And then what do we do with that information? We can take it to the epidemiologist. Rapid is used when the microbial population is unknown in a sample. Let's just say sample from a river. And what we do is we use a whole bunch of different little pieces of DNA called primers that we manufacture on a machine that then are going to stick to the DNA that's in the sample at a particular spot. And then the enzymes that build the DNA that we throw in are going to build off of that spot and make many copies of the DNA that lies between those primers, And then we can analyze it and ask the question, what does the sequence of this DNA compare to from all our previous information? Okay. In hybridization, we take a piece of genetic material that we either build or that we manufacture using enzymes and source DNA that come from, say, bacteria or plasmids. And then we generate large amounts of either dye tagged or radioactive probe that will hybridize to the target, meaning that it'll hydrogen bond with it because the base sequences are pretty complementary. Okay. And that indicates that that information is present and that that probably tells us what the microbe is. Okay. All right. Let's take 15 and we will come back at 2 o'clock. Okay. All right. We're back.
Yeah.
All right. We had left off talking about genetic methods to determine what kind of bacteria, what kind of pathogens are growing in your sample. And one of the methods you can use is something called hybridization. All right. And all you need to know about this is that you can make pretend pieces, well, not pretend, they're real pieces, but artificial pieces of DNA with a machine, and you can attach them to a dye that glows when you hit it with a laser beam. All right. And then you can mix those pieces of, they're called primers, with your sample. All right. And then the only places where those primers will stick is to the information that's coming from bacteria that you think are causing the problem. All right. So you whip these up in a machine, you have them ready, you mix them with your sample. They stick where they're supposed to on the slide. And you can put that slide under a special microscope that makes a laser beam that hits the whole slide, but where the probe is stuck, it'll glow a particular color. And you're able now to see where your bad guys are. And you can also do this in a test tube, a little plastic tube. This is called an Eppendorf tube. Okay. And you just mix your probe, add your probe to your sample. There's your probe, right? And your sample comes from the patient sample. Okay. And then you heat it up and then cool it down. And then what will happen is that if the probe sticks to genetic material that's in your sample, it'll turn a particular color when you hit it with a laser. Okay. And if it doesn't stick, you won't get that color. All right. So you can do this with fluorescence. Fluorescence uses dyes and lasers. Okay. Or you can do this with radioactivity. That exposes X-ray film. Okay. This is another method where you can take DNA from your sample and you can run it through something called a gel. All right, what is a gel? Think of it like Jell-O. But it's made from plants, particularly seaweed. Okay. And what happens when you apply a current to it while it's sitting in a container that has buffer, right, is that the current will pull the DNA that you've isolated from your sample in a particular direction. All right. And the bigger the DNA, the tougher it is for it to move within a certain period of time. And the smaller the DNA, the easier it is for it to move, right? So you separate the DNA molecules by their size, and then you can probe them, all right? Or you can look at them directly under UV light and see the size of the DNA molecules. And that can tell you the identity of your particular pathogen because of data that they already have. They can do the same thing with what they call ribotyping, okay, where we take ribosomal RNA from your sample, including RNA, ribosomal RNA from the pathogen. And then they can use PCR to amplify the ribosomal RNA. And then they can compare that to data already collected in big old databases, okay, computers, okay. You can use something called FISH, where you have different dyes for different types of ribosomal RNAs attached to primers. And then depending on what colors light up, you can tell what kind of organism it is, okay? So just another way to do it. This is a look at how FISH works. Come on, give it to me. Let's do this. Let's do what we do. Control and move down. Let's do this. There we go.
Situ hybridization is used to detect the physical location of a gene on an intact chromosome. In situ is Latin for in place, and in this procedure, the chromosomes are adhered in place onto a slide. The most common method of in situ hybridization uses fluorescently labeled DNA probes. This technique is known as fluorescent in situ hybridization, or FISH. To perform FISH, first cells that have been arrested in metaphase are treated to make them swell, and then they are fixed onto the surface of the slide. This also fixes their chromosomes onto the slide. Next, the slide is treated so that the chromosomal DNA is denatured into single strands. Then special DNA probes are flooded onto the slide. DNA probes are small pieces of single-stranded DNA with a sequence from the gene of interest. Probes are able to hybridize only with their complementary sequence, which will be at the site of the gene of interest on a particular chromosome. The DNA probes are allowed to hybridize with the denatured chromosomal DNA, and any excess probes are washed away. Either the DNA probes themselves are fluorescent or they are chemically modified to allow fluorescent labels to attach to them, so that when fluorescently labeled molecules are added to the slide, some are able to bind to the probe DNA that is bound to the site of the gene on the chromosome. Excess fluorescently labeled molecules are then washed away. The slide is then viewed using a fluorescence microscope. The fluorescently labeled molecules reveal the physical location of the gene of interest.
So it's just a way to see the information on the actual slide. Okay. Sort of a clever way to detect it. And like I said, you don't need to know the exact ins and outs of how you do it. You just need to know that you're able to stain and tag the information that says that that pathogen is in there. Okay. That's really the most important point. All right. All right. We'll talk about some different methods of detection. Okay. Talk about serology and ELISA, what they're all about. Okay. So the bottom line with this, all right, all of these immunological methods is that you can find a tool to attach to a molecule that your pathogen has. Okay. And that tool is an antibody. Okay. Antibody attaches to one antigen, which is a molecule on the surface of the pathogen. It's on the pathogen. So that's the only place he's going to stick. All right. And then the antibody can be attached to a dye that glows when you hit it with a laser beam, just like those probes that we saw in things like fish. Okay. Or he's going to have an enzyme attached to him that'll make a color reaction when you add a certain chemical to the slide or to the tube. Okay. So an example, right? You can test serum from blood, right? To see if your bad guy is in the serum, right? Or was at one point in the serum. What'll happen is that you'll look at the interaction of antigen and antibody, and that lets you determine whether that pathogen was ever there. Urine, cerebrospinal fluid, whole tissues, and saliva can be analyzed for the presence of a particular antibody, and that allows you to determine the immunologic status of the patient, meaning have they been exposed or not exposed? Okay. Oh, stop it. Sure. So exposed or not exposed. Okay. If they're exposed, they will have antibody. They're not exposed. There won't be any antibody. Okay. And so you mix antigen from a particular pathogen with serum from your patient. And if you have binding, all right, you're going to get a particular color when you either hit it with a laser or when you mix in a particular chemical. All right. And that color will tell you, right, that the patient has antibody. Okay. One color if the patient has antibody, a different one if it doesn't, if the patient doesn't. All right. Okay. You can also ask the question, what is the identity of a particular microbe, right? And so you can have antigens that are on the surface of the pathogen, and then you can have antibodies against all these different types of antigens, right? And so you can test for the presence of an antigen, say, to pneumonia, or an antigen to, say, a staph infection or something like that. And if you get binding and a color change, then you know that that organism is in the patient somewhere causing trouble. Okay. So here you're detecting the antigen. Okay. So this would be active infection. Active infection, right? And here you're detecting antibody. Okay. And this would be a past exposure. Okay. The most effective serological tests are very specific and very sensitive. Specificity means that it only is going to attach to a particular target molecule and a tiny place on that target molecule. And sensitivity means that you can detect tiny amounts of that antigen or that antibody, and every person who's been exposed will show up as a positive reaction. So how is it that we see the color change? Well, we want to make it visible to the naked eye, all right, or at least to seeing it through a microscope. And what happens is that when the antibody binds the antigen, it can clump the pathogens that bear the antigen, and we can see them microscopically, or in some cases, even with the naked eye, all right? And then smaller interactions can use dyes or fluorescent reagents, okay, right, or enzyme reactions. Okay, so let's take a look at agglutination. Let's see if it's going to give us the high sign here. Probably not. All right, so we're going to do the same trick again. Let's go in.
A number of in vitro diagnostic tests are based on the specificity of interaction between antibodies and antigens. When soluble antibodies react with insoluble.
Hold on. Just a little share.
A number of in vitro diagnostic tests are based on the specificity of interaction between antibodies and antigens. When soluble antibodies react with insoluble particles, such as bacteria, the antibodies link the bacterial cells together, forming an agglutinate. When certain viruses, such as the measles virus, are mixed with red blood cells, the virus particles react with structures on the surfaces of the red blood cells, causing hemagglutination. If the patient has measles, the serum will contain antibodies against the measles virus. If the patient's serum is mixed with measles virus and red blood cells are added, hemagglutination will not occur. Antibodies reacting with the virus particles prevent reaction between the virus and the red blood cells. Agglutination tests can also be used to measure antibody titer. In the tube test, a specific amount of antigen is added to a series of tubes. Serial dilutions of serum containing antibody are then added to each tube. The greatest dilution of serum showing agglutination is determined, and the reciprocal of this dilution is termed the antibody titer. When soluble antigens such as proteins react with soluble antibodies, they form a precipitate. If the antigen solution is layered on top of the antibody solution in a test tube, a precipitation ring forms in the area where the two solutions make contact. The precipitin reaction also occurs when antibodies and soluble antigens are mixed in the proper proportions. The antibodies link the antigens together to form a precipitate that settles out of the solution when it becomes sufficiently large. Immunoprecipitation increases as more and more antigen is added. When the optimum ratio of antigen to antibody is present, called the equivalence zone, the maximum amount of precipitate is formed. If more antigen is added, smaller complexes and therefore less precipitate is formed. A graph of the amount of precipitate formed versus the amount of antigen added shows the amount of precipitate increasing until the zone of equivalence is reached. As the amount of antigen added becomes in excess of the antibody, the amount of precipitate decreases.
So what you're doing is you're detecting the presence of either antibody or antigen as a result of the fact that they bind to each other very tightly and very specifically, okay? And you can attach the antibody to a dye or to an enzyme before you use it if you're detecting antigen, right? And if you're using antigen, you can do the same thing with the antigen, right? And that allows you to detect when they attach to each other, okay? All right, so back out we go. Here, new share. Okay. A titer is just the amount of something in a sample, okay? You can titer phage, you can titer virus, and you can titer antibody, okay? And it's determined by diluting the sample and throwing in equal amounts of target. And we try to figure out here the highest dilution of sample that produces agglutination, right? The more serum sample that can be diluted and still react, the greater the antibody titer, right? And so we can use this to do all kinds of tests. We can look at autoimmune disorders, which are antibodies against your own tissues. And we can also determine whether you've been exposed to certain diseases in the past, such as tuberculosis, okay? Serotyping is a way to classify bacteria or really any pathogen into smaller categories that are called serotypes. And it uses the presence of special molecules on things like the capsule or the cell wall or the flagella of the bacteria, if we're looking at a bacteria, in order to determine the serotype. We've used this in identifying different Salmonella species and the basis for the differentiation of pneumococcal and streptococcal serotypes. Okay. The quelling test involves a precipitation reaction against polysaccharide antigens that are in the capsule. Okay. So here we are detecting the pathogen, right? Detecting the pathogen. Okay. And so this is something that we would find in a patient in an ongoing infection. In a Western blot, what we do is that we extract the protein from a sample, and we can run that through a gel, just like we can run DNA through a gel, and separate it based on its size and its shape. And then we can take that gel and we can transfer those proteins to a piece of paper, filter paper. We do that with an electric current. And then we can throw particular antibody at that paper that's attached to something that will produce a color. All right. Often it's an enzyme. Okay. Or it could be another antigen that another antibody will react to that has an enzyme on it. And then we either hit it with a laser beam or we throw on a particular chemical, and that part of the paper will change color. And that tells us that that protein is there in the sample and lets us indicate that that particular pathogen is there, right? So here we are finding protein from the pathogen. Okay, finding protein from the pathogen, and the antibody is our tool. In immunofluorescence, we use antibodies that are attached to dyes that glow when they're hit with a laser, all right? So we throw the antibody and dye complex onto a slide, all right? Or we could throw it onto a Western blot in some cases. And then we hit it with that laser, and the areas that have the antigen are going to glow because the antibody is going to stick to it. Okay. So if the complexes form, they stay bound, right? And then we can see them through a microscope with a laser if we're looking at a slide, or with our naked eye if we're looking, say, at a blot. Okay. So again, we are detecting antigen on pathogens in an infection. Okay. Indirect testing are where antibodies recognize constant regions of antibodies in the patient. Known antigen is added to the test serum, and the binding of the fluorescent antibody is picked up with a laser that causes the dye that's attached to the sandwich of antigen and antibody to glow. Okay. So the idea here is that you have patient antibody. We'll draw that down here, patient antibody in red. Okay. And then we have the antigen, and we'll do the other blue. Okay. So there's our antigen. Okay. Okay. And then we throw on known antibody. Okay. Okay. That will have a dye attached to it. This is attached to the plate. Okay. This is the antigen, right? That we supply. Okay. And this is the other antibody that we supply. Okay. And this is an antibody from patient. Okay. So this is for an ongoing infection. Okay. Sorry, not ongoing, past infection. Erase that. Okay. Other methods include radioimmunoassay, where antibodies are going to have a radioactive label attached to them. And we can use this to detect the presence of a particular molecule in a sample. And then ELISA, which uses an enzyme-linked indicator antibody to see the antigen-antibody reaction, okay? And so that's sort of like what's going on in the previous slide here, okay? This could be an ELISA type test. Okay. ELISA uses this same principle, okay? Except the wells are tinier, right? And we use a machine to scan the wells to look for the color change that we pick up when the dye is attached to something that's attached to the antigen, the patient's antibody. Okay, so that's the idea. Okay. An indirect ELISA is going to pick up microbe-specific antibodies in the patient. A known antigen is attached to the surface of a well and mixed with unknown antibody. An antigen-antibody complex gets formed. An added indicator is an antibody that will bind to the antigen, makes the color change. It's a common test for antibody screening, HIV and rickettsia. Okay, sometimes you'll get a false positive. We use a Western blot to back up the data. Okay, in a direct ELISA, known antibody is attached to the well and incubated with unknown antigen, and then antigen-antibody complex forms, and then we have an indicator antibody that we throw in, and the color develops in those wells. Okay, just like we diagrammed 2 slides ago. Alkaline phosphatase can be used for visible light, okay, that can be quantified by a machine or on a photo, okay. There are computer chips that can pick up tiny changes in current that happen when antigen-antibody complexes are formed. So that's another way of determining whether you've got a reaction. But you can see here some examples of how this works, right? The areas in the plate down at the bottom, okay, where you can see here yellow wells. Indicate patient antibody. Okay. So again, this is for past exposures. Okay, the next thing is complement fixation. So let's hop over and get that.
The complement fixation test is used to test for the presence of specific antibodies in a patient's serum. Patients differ in the amount of complement in their serum. To standardize the complement fixation test, the first step is to heat the serum to destroy the patient's complement. A measured amount of complement and antigen are then added to the serum. In patient A's serum, the complement is fixed by reaction with the antigen-antibody complexes. In patient B's serum, there are no antibodies to complex with antigen, and the complement remains free. To determine whether complement has been fixed, sheep red blood cells and antibodies against sheep red blood cells are added. In patient A's serum, the complement is already attached to the antigen-antibody complexes in the serum and cannot induce sheep red blood cell lysis. Consequently, the red blood cells settle to the bottom of the tube. In patient B's serum, the complement is free to react with sheep red blood cells antibody complexes, resulting in lysis of the sheep red blood cells. The serum turns a pink color as the red blood cells lyse.
So very straightforward, right? You either get clearing in a particular color or you don't, right? If you get clearing in a particular color, that means the patient hasn't been exposed, all right? And if you don't, that means that they have, okay? Very straightforward way to do the test. All right, moving down. In vivo tests use the same idea as serological tests, but the antigen and antibody is put into the patient, and we look for a reaction in the patient. Okay, an example is the TB test, where a tiny amount of antigen from Mycobacterium is injected under the epidermis of the skin, and then if you get an induration, which is a raised red bump on the skin, that means they have antibody against that Mycobacterium antigen, which means that they've been exposed in the past. Okay, so again, this is an example of past exposure. Okay, past exposure. Okay. Okay. We'll talk about different culture methods. The gold standard of diagnosis is to grow the pathogen in a culture and ID it outside of the patient. Okay. What happens if you ID something that happens to be in your sample that isn't the pathogen, right? In addition, culturing takes some time, right? A day and a half in some cases. And a lot of infections involve more than one pathogen, right? They're polymicrobial. Example is a blood infection. Identifying the cause of septicemia can take between 18 to 24 hours using traditional methods, but the time frame is 6 hours. If a patient responds to a broad spec antibiotic, the prescription is not rewritten for a more appropriate drug. If the patient doesn't improve, the delay is fatal. So a more effective technique was developed, okay? one of these techniques is called a chip, okay? And what a chip has is, a chip is just a piece of glass, right? Tiny piece of glass, sort of like a cover slip almost. And then within that piece of glass, let's pick another color here. With gray. No cyan, fine. Are areas on a grid, right, that have probes, DNA or RNA probes, usually DNA probes, to specific antigens, right? And when we throw our sample on there, okay, the genetic material from that sample can bind some of these probes. And if it binds some of these probes and we hit it with a laser, a particular color will be generated. If it doesn't bind the probe, a different color will be generated. So the colloquial for that is generating what's called a heat map. Okay. And the areas that are hot are the areas that have probes that are what are called homologous, meaning the same information is there as is in your sample, and you can determine what your sample is. Okay, so again, we're looking at the genetic information, and this is pretty quick. Okay, so we throw this under a device that has a laser. The laser hits the chip glows. We take a picture of the chip, and then the computer analyzes the colors and then matches it against the data on the chip. And we're able to determine in a single test what's in there. Okay. We can now take the sequence of entire genomes of organisms in a relatively short time. And this is called next generation sequencing. Okay. And this allows us to get a lot of information very rapidly that we couldn't produce before with other methods. Okay. So it's fast and it's comprehensive, right? Fast and comprehensive. Okay. And in addition, it's accurate. Right. So that means that we get to the heart of the problem very, very quickly. Okay. In mass spec, what we do is we take protein from a sample and we blow it into little pieces using an enzyme that digests protein, usually trypsin. All right. And then we make those little pieces of protein fly across a detector, and the detector is able to determine what's called the time of flight. That's what TOF is for. Time of flight. Of that little piece of that protein from the sample to the endpoint. Okay. And then we can use that information to determine what proteins are in your sample. Okay. So it finds proteins in your sample. Put that here. And IDs those proteins and compares them to known proteins. And then you can determine what pathogen is present. Pathogen is in your sample. Okay. Like I said, you don't need to know the specifics of how this works. Just know that it's able to figure out what proteins are in your sample, compare those proteins to information that we already have, and then identify the organism. Okay. We can use MRI, CT, and PET scan also. Okay. The advantage of the modern methods is they're way quicker. All right. And so they're way more effective. And they can allow us to deal with infections that happen so rapidly and need to be treated so quickly that we don't have time for staining and culturing and all the other stuff that we use traditional methods for. Okay. So it's faster, it's more accurate, and the cost is pretty similar now. All right. So this is a lot of what we're going to when you ship a sample out to what they call a core lab. All right. So there's labs all around town that take samples from all the different hospitals, and then they shoot the information back to the hospital almost immediately, and they're able to identify real quickly what's going on. Okay. What kind of infection do you have? What drug should I pick to treat it? Okay. Now, as far as lab goes, all right, there's two things that you'll be doing. All right. There's a 5-point exercise on Connect that deals with looking at pond water. Okay. And I can show it to you here real quick. So let's go to Connect. All right. And we'll go to our home page. Sign in. Stop it. Sign in. So all this other stuff. Okay. Go down here. Right, so close that out. So, screen. We want these here. All right. So we go into our section. All right. You'll see something called pond water wet mount. All right. And what you want to do is you want to go into that and you want to do that exercise. Okay. And Connect will score it for you. You have unlimited attempts. Okay. And it's pretty simple. All right. And then I'll collect those points and score them. And then the other thing you're going to get from me is a water pollution lab that I'm going to send you. All right. So I will send this to you. You'll do it using the instructions that are in the PDF, and you'll send that back to me in the email. And then I'll include that in your scores as well. Okay. So two things, right? This is just looking at pond water and seeing what you typically find in it. And so you're going to see all sorts of different things like bacteria, protists, and maybe even some worms. All right. And you'll identify them, and that report will file and grade, and you'll get those points. And then you're going to turn around and with some chemical tests on a different website, you're going to identify what are called coliform bacteria, which are bacteria that can show up in drinking water that's been contaminated with fecal material, right? As you know, feces contains E. coli, right? And that's where coliform bacteria, if they're in the feces, can get into fresh water if there's been contact, and you can determine how many coliform bacteria are in the drinking water and whether it's safe to drink, okay? And so that activity will show you how to do that test on a website online, and then you'll fill out that report, and then you'll shoot that back to me, and that'll be the other 5 points, okay? So not too complicated, all right? The good news so far is everybody's doing pretty well. in the course is I've updated the scores so you can see those now on Connect, not on Connect, on Compass or on your web portal. All right. And as you use those codes and complete the remaining assignments, I'll update those as soon as they're submitted and they're done. Okay. If you have any trouble with a code, let me know so that I can get in touch with the rep so I can find out what's going on. In some cases, the rep may just be able to take and convert you directly to a full-time student at no extra cost. Okay. So we got these codes for nothing. In addition, if you look on the home page, you should see the ebook, right?
Yes.
So here you can go and you can read the ebook and get the background information for what we've covered in lecture in sort of a basic format. Okay. So there you go. Everything that you need in one spot. Okay.
I have a question.
Yes, ma'am.
I couldn't open the lab homework last week.
Okay. What program did you use to open it?
You said to get Adobe, right?
Adobe Acrobat Reader. Yeah. Did you
Yeah, I
download
can't-
and install that?
I did, but I couldn't open it up.
Okay. Are you using
Can
a
you send
PC?
me another email? Maybe
I can send you another email. Are you using a PC
Okay.
or a Mac?
PC.
Okay, so on PC, what you have to do is you have to right click on the file after you download it in the email,
Okay.
and then you have to pick Adobe Acrobat Reader as the application to open it.
Okay.
And then
I'll
it'll
try.
open. Try that. Okay, but I'll resend it. Okay,
All right. Thank you so much.
welcome. Any other questions before we bump out? We good? All right.
Yeah. All right.
I'll send you the Zoom link for the Friday tutoring. If you want to come
Okay.
in, you want to stop in. Or if you need a different session, let me know and we'll plan a different session. Okay. Or maybe a
Okay.
time that works better for everybody. Maybe we could all come if you want, and we could work on that. So let me know. I'll supply you that information. I'll make those adjustments. And other than that, I will see everybody next Wednesday. Okay.
All right. Thank you.
Thank you.
Mhm.